Hello, everyone. Today, the group will be presenting our project on how chromatin accessibility shapes gene expression differences between healthy and diseased tissue in diffuse large B-cell lymphoma, or DLBCL. Our guiding question was, how does chromatin accessibility shape gene expression differences between healthy and diseased tissue in DLBCL? And we focus on this because while genetic mutations in DLBCL are well studied, the way DNA packaging and accessibility contributes to the disease is not fully understood. DLBCL is the most common type of non-Hodgkin lymphoma. It's very aggressive, it grows quickly, and it has a 5-year survival rate of about 60%. Malignant B cells are larger and more dispersed than normal, and patients are often presented with a wide range of symptoms, which makes it difficult to diagnose, and it also makes treatment very challenging. Traditionally, research has looked at DNA mutations, but there's been a lot fewer studies that look at how the DNA is packaged and whether regions are open or accessible, and how that can drive cancer progression. In DLBCL, it's still unclear exactly which regions change accessibility and how that impacts gene activity overall. Research objectives. To address the gap that was discussed in the previous slide regarding chromatin accessibility, we developed 3 objectives. The first is that we were going to identify genome-wide differences in chromatin accessibility between healthy and diseased tissue. The second was we were going to characterize the 3D chromatin structures linked to abnormal gene expression. And then the third is we were going to test the functional significance of specific regulatory elements that drive dysregulation in DLBCL. To do this, we're going to take a multi-step, multi-omic approach. First, we're going to map open and closed chromatin regions with ATAC sequencing. Then we're going to integrate that data with 3D genome mapping from Hi-C and gene expression data from RNA sequencing. Finally, we're going to use CRISPR tools to directly test whether those altered regulatory elements are controlling oncogene expression and cancer cell behavior.
Okay, I'll be presenting experiment 1, which focuses on how chromatin accessibility contributes to gene expression in DLBCL. We're going to be using a method called ATAC-seq that allows us to map open and closed chromatin regions across the genome. Open chromatin have regulatory regions such as promoters or enhancers that control gene expression. So these regions can lead to the activation of cancer-causing genes or genes that help cancer proliferation. In the experiment, we're going to be using healthy B cells and DLBCL cell lines to minimize variables here. We'll isolate the nuclei, keeping the chromatin intact, and treat them with a hyperactive enzyme called TN5 transposase. This enzyme cuts the DNA that's accessible and tags it with adapters for sequencing. In the first picture on the top right, there's a graphic of what the enzyme would do, and the adapters are going to be the blue and red marks on there. The resulting fragments are amplified using PCR, and libraries are created to generate FASTQ files that have the sequencing reads. Those reads will be aligned with the human genome, and the peaks are going to be called using a program called MAC-S2 to identify the chromatin peaks in the data. The peaks in the software represent the chromatin regions that are going to be accessible. In the second picture on the bottom right shows what an example of the output of what MAC-S2 would show. By comparing the peaks of healthy and cancerous B cells, we can identify which chromatin regions are open and closed in DLBCL. The estimated timeline for this experiment is about 8 weeks, including experiment and data analysis. The sample gathering can take around 10 hours to complete, but this means the quality of the sample needs to be precise. The data analysis would take longer once the sequences are aligned and the peaks are called. In essence, ATAC-seq gives us a map of the regulatory regions and how changes in chromatin accessibility can contribute to disease progression. This experiment lays the foundation for the next experiment in our proposal. Once the regions are discovered, we can learn more about the structure.
Hello. So we're going to talk about experiment 2. We're making 3D chromatin structures using Hi-C as a genomic tool and RNA sequencing. Chromatin accessibility is based on spatial organization between loops, and Hi-C is a genomic tool that's used to map out these 3D architectures of the genomes. Hi-C is a chromosome conformation capture carbon copy, and it identifies TADs, which are topically associating domains and chromatin loops. For a sample size, we're using CD19 plus B cells taken from frozen biopsies of healthy donors and DLBCL donors. The method we're using is in situ Hi-C. We're preparing coding libraries for the tumors and the control samples, and using RNA sequencing to extract the RNA, obtain that gene expression, then pass it through Hi-C Pro, which will make some matrices from the raw sequences. Then it will pass through the juicer, which will make a 2D model that now interacts with high glass. That will make a 3D model that is able to be shared for more visualization and manipulation for later research. And that's to identify the TADs and then compare chromatin structures. What we expect as a result is identifying the oncogenes and chromatin loops that are doing this type of gene expression, and then linking the 3D model to the gene malfunction that happens with DLBCL samples compared to the healthy donors. So in the timeline that we are expecting to complete this is about 12 weeks.
I'll be presenting experiment 3, test functional significance of regulatory elements with CRISPR interference and CRISPR activation. The techniques used in prior sections, including ATAC sequencing, RNA sequencing, and Hi-C, are used to show correlation when identifying regions where there are changes in gene expression in diffuse large B-cell lymphoma. For my portion of the research, I chose to discuss the potential of using CRISPR interference and CRISPR activation to test causality and see if by changing accessibility to specific regions, if it would cause changes in gene expression. So I chose 3 enhancers based on some criteria from the other sections, including more open and DLBCL from ATAC sequencing, physically interacting with oncogenes from MYC or BCL6 from Hi-C, and association with higher expression of genes from RNA sequencing. So for mine, I would utilize CRISPR interference to repress the regions to see if oncogene expression decreases, and then follow up using CRISPR activations to activate closed enhancers to measure if the expression will change. Finally, we can further monitor any changes in gene expression and analyze cell behavior using PCR and RNA sequencing. If the expression drops, then we know it and the enhancers are blocked, we have evidence that chromatin accessibility can regulate gene expression in DLBCL. To further go over concepts, I use the article Kasperzik et al., and they had a similar approach in using B-cell lymphomas by focusing on immunoglobulin heavy chain super enhancers that controlled gene expression in cancer genes like MYC and BCL2. Based off of their reports, it showed that blocking those specific regions reduced cancer cell survival. Based off of timelines outlined in similar experiments, we can estimate that this experiment can take roughly 6 to 8 months to complete.
For our hypothesis and expected outcomes, we expect to find that certain enhancers and promoters, like the ones listed there, are abnormally open in DLBCL. We also expect to see disrupted 3D genome organization, such as oncogene enhancer looping and TD boundary changes that are correlated directly with overexpression of key oncogenes. Functionally, targeting these elements should alter gene expression and potentially reduce overall cancer growth. For broader impacts, by combining ATAC sequencing, Hi-C, RNA sequencing, and CRISPR, our project not only identifies changes in chromatin accessibility, but it also shows how they connect to 3D genome structure and function. This approach could ultimately be applied to other blood cancers and help integrate chromatin architecture into personalized medicine. In summary, our research aims to bridge the gap between genetics and epigenetics in DLBCL by showing how chromatin accessibility and structure influences cancer progression. This work could highlight new therapeutic targets and inform broader strategies for cancer treatment. Thank you, and I'm happy to take any questions at this point.